Taking a trip down memory lane back to 7th grade, or at least what I should have learned in 7th grade. But for me, this is something that I learned in EMS, and it really stuck with me. So membrane transport. Before I start talking about the two main mechanisms, let's talk about the two types of directionality we can have for membrane transport. Ugh. We have symporters and then we have antiporters. So symporters, that should be an N by the way, not an M. Symporters. Synonymous, right? So they're both headed in the same direction. For antiporters, it's anti, right? They're headed in opposite directions. Hopefully you can see those are arrows. I have this pen set way too small. But anyways, there's two other types of, or two, I guess, means of which we can have things transporting, and this is based off of whether or not we're using energy. One of them is called active transport. And as you can imagine, we're using energy in the process of active transport. And the other one is known as passive transport because we're not using energy directly to cause this transportation process. And for passive, there's really just two subcategories of passive transport that we would need to talk about. And I'm going to go ahead and just draw them really large like this with the arrows denoting them. I can't draw in straight lines to save my life. But in passive transport, what's happening is whatever it is that we're moving is going to be moved from a high concentration to a low concentration, right? Think of it like dropping a pen or dropping a ball or dropping anything. You're not putting energy into making it fall. It just passively falls on its own, right? So for the two categories, we have facilitated diffusion and then simple diffusion. For simple diffusion, this is really two types of molecules that can undergo. Simple diffusion is where things just simply move from one concentration to the next. And because these things are either going to be very, very small polar molecules or they're going to be nonpolar molecules. Nonpolar molecules that are totally cool with passing through the fatty acid tails, or in this context, the small polar molecules that can just kind of wiggle their way through the fatty acid tails. These would be the things that do simple diffusion. So to give an example, and the first example that I'm going to give, you're going to think that I'm being a misnomer here, but yes, this is true, that water does move across the cell membrane by simple diffusion. It doesn't move in a large amount at a very fast rate through simple diffusion. So our cells have had to use mechanisms, specific proteins involved in moving water. But water does move through simple diffusion, right? Also, anything that's usually tends to exist in the gaseous state, assuming it's not too large, can diffuse through simple diffusion. We don't have a carbon dioxide channel. Protein that we need to use. We don't really worry about that. I'm trying to think of any others that are involved in this. Certain gases, it's kind of weird. Gases, if they're small enough, can go through there, but other ones that also are small but exist in quantity are used as receptors, but that's a different game altogether. For nonpolar examples of this, things that could come through here, this would be steroid-based hormones that we had talked about already. I'm just going to say steroids, anything that's lipid soluble. And then for facilitated diffusion, this is, hands down, this is the fastest transport system ever. Facilitated diffusion is faster than active transport, and it's faster than simple diffusion. So what's happening in this is just with this, and the concentration gradient though. So basically all we have is something moving down a concentration gradient with the help of a transporter. With the use of a transporter. And this transporter, more often than not, is going to exist as a channel. A channel that's just going to open things up and let things passively flow from a high concentration to a low. Didn't mean to circle this in white, but cool. So whereas passive was moving from a high concentration to a low, the concept of dropping a pen or dropping a ball or dropping a MacBook Pro that's a piece of shit off a laptop, off of a building, the active transport, we're actually having to use energy in the process because in this we're moving up against our concentration gradient. So we're going to start off in a very low concentration and we're going to be moving to areas of high concentration. For active transport, we have actually two subclasses of this. There is primary active transport, and then there is secondary active transport. So in case this wasn't obvious, we are in fact using energy. What is the most common energy molecule in cells is ATP. So there are two types of examples that I'm going to give is P-type ATPases and then ABC, ATP binding cassettes, right? So example of a P-type primary active transporter would be the sodium, potassium. P-type is this is where the P comes in. It's an ATPase. So one of the things you may want to take note of is if we're involved in the use of ATP. For most of the time when we have this, we're always going to use a phosphoryl aspartate, otherwise known as aspartic acid, but remember, physiological conditions, it's always going to be deprotonated. And then for ABC binding cassettes, these are, there's really just two, I guess, subunits that we could talk about here, two, I guess, categories of dimerized subunits. So two of these, it's going to have two membrane domains, and then two of it are going to be the actual ATP binding sites. So I don't know if you guys have ever heard of iTunes University, but Randy Sheckman out at Berkeley, who just recently won a Nobel Prize, gives one, and in his he talks about ATP binding cassettes, and he talks about their role in cancer, and that cancerous cells have way more advanced ABC transporters, and they have way more powerful and effective transporters, but also that oncologists give drugs that inhibit the activity of ATP binding cassettes. And for me, that seems kind of stupid to do if you have an increased gene expression of this stuff. This is involved in moving toxic chemicals out, like we said earlier, and so this is going to be kind of, in my opinion, counterintuitive, because for every one cell in your body that you're going to kill, or every one cancerous cell that you kill, you'd end up killing 5 of your own cells. I don't like those odds, so that's just my opinion, though. So yeah, this is ATP binding cassettes are carrier proteins, or they use carrier proteins involved in them. Carrier proteins, and these are both associated with multi-drug resistance, MDR, cystic fibrosis. These are just involved in pumping things out of the cell. I'm just going to say that it pumps a lot of stuff out of the cell. I'm keeping things abstract at this point. For secondary active transport, this is, I don't know why this is so difficult for some people to imagine. When I'm done with this, I'll draw a picture of it. Secondary active transport, if you have, say for example, the P-type ATPase, which is the sodium potassium pump, and I'm going to make the assumption that you know what that is. This is a 3,000 level biochemistry course. I understand that it's not for majors, but still, sodium potassium pump, you should have learned about that in 8th grade biology. We have sodium being pumped out of the cell and potassium being taken into the cell. So what this is this is a new concentration gradient that we have formed. Concentration gradient that we have formed from the primary, from the action of the primary. So I'm just going to say from the primary transporters. Primary transport mechanism. And this usually tends to be a symporter or an antiporter. So I'm going to say that usually tends to be symporter or an antiporter.